This video is ECR's MAP Kinase. A phosphorylation cascade is shown here. This is a sequence of events where one enzyme phosphorylates another, which causes a chain reaction leading to the phosphorylation of many proteins. I like to think of this as a domino effect, so I've got a picture up here. Basically, it's like if one knocks one domino over, for example, one signal molecule binds to a receptor, then a whole cascade of events will take place, one leading to the next to the next. And so this idea of a domino effect is kind of like a cascade. And so as you can see in this drawing here, you've got one activated molecule, which will lead to the activity of one kinase, and that will add another phosphate group to another molecule, in this case another kinase, which then can phosphorylate another protein due to its activity, and kind of goes down the line. So this is the phosphorylation cascade here, is a series of kinases which are activated one from another, and transfer of phosphate groups, typically from ATP, is what's going on. Finally, so what we see kind of here is, so if we knock over just one domino here, then that can lead to a whole series of events like we see over here. And so basically that's what I think of as the phosphorylation cascade. And we've talked about that before, is a distribution of signaling through pathways. Phosphorylation cascades are incredibly common, and this happens to be a very efficient way to transmit a signal. So now we'll talk about the MAP Kinase Cascade, or MAP Kinase module, as your textbook refers to it. And basically the deal is that RAS, in its activated form, over here when it's bound to GTP, will activate a MAP kinase. And so that's an enzyme which can then start off this cascade. So MAP kinase on its own, let's talk about what that stands for. So MAP kinase refers to the mitogen activated protein kinase. And so basically mitogen, notice that sounds a lot like mitosis, and gen being generating. So these kinases typically are going to lead to cell division and cell proliferation. In other words, mitosis. And the way that they allow the signal to be communicated is through kinases. So if we start at the beginning, we start with activated RAS, in other words, RAS GTP, and that activates an enzyme called MAP kinase, which is kind of a silly name, but the way that this works out is that the ones with the most kinases are early on, and then the ones with the least kinases are on later. MAP kinase will phosphorylate MAP kinase, which is shown in blue over here. And again, this is through the transfer of a phosphate group from an ATP molecule. And then a MAP kinase will activate a MAP kinase. Then the MAP kinase will actually trigger a variety of different effector proteins. And you can see that over here that there's protein X, there's protein Y, there's different transcriptional regulators, and it's a little bit blocked out here, but basically this can result to changes in protein activity and changes in gene expression. Oh, and by the way, this is figure 16-34 in your textbook. So the effector proteins can go on to a variety of different roles, including cell growth, making a larger cell, cell proliferation, which would be cell division, like mitosis, cell survival, also a key part of cell lifespan, and then cell differentiation. And that's a process during development that allows one cell to take on different characteristics from another. Not super critical for our concepts in this course, but just so you know that this is a developmental process. And one more thing as a reminder, we just picked up here with the activated RAS, but remember where RAS came from. So activated RAS comes from receptor tyrosine kinase signaling, which has been activated. Finally, just to talk a little bit more about this kinase versus kinase, I think this is a way to kind of show that these different proteins are all related to each other sequentially. And essentially, the MAP kinase is not that it's phosphorylating at more locations, it's just that a MAP kinase will phosphorylate a MAP kinase, and then the MAP kinase will phosphorylate the MAP kinase. And so it's a little kind of, I don't know, silly or something, but it's a way to keep track of these molecules and how they work sequentially in a pathway. This is a core pathway that's used in many different organisms, across model organisms and through humans, and it's, as you can see, a core part of the life cycle of animals.